Let's talk about a technology that is literally rewriting the code of life itself. I'm talking about gene editing. And look, this isn't some far-off science fiction concept. It's happening right now, and we're only just beginning to see what it can really do.
So I want you to imagine your DNA for a second. It's this massive 3 billion letter blueprint for your entire body. Now what if there's a tiny spelling mistake in there, a single typo that causes a serious disease? What if we had a biological find and replace tool that could go into that code and just correct that one little error? Well, that's the incredible promise of what we're diving into today.
So how on earth is something like that even possible? Well, it all comes down to this absolutely remarkable tool that scientists have managed to borrow from nature. So let's get into it and meet these so-called genetic scissors.
Okay, so this tool is called CRISPR. And the wild thing is, it wasn't just invented from scratch in some high-tech lab. It was discovered. And get this, it was discovered in bacteria. Yeah, researchers, some of them just studying microbes in yogurt, figured out that bacteria have this ancient built-in immune system they use to fend off viruses. They literally chop up viral DNA to protect themselves. And scientists, in a stroke of genius, figured out how to adapt that natural system to target and tweak specific genes in us with unbelievable precision.
That sounds amazing, right? But how does it actually work once it's inside a cell? The whole process is actually pretty elegant, and you could really boil it down to 3 main steps. So first up, you have a molecule called a guide RNA. Think of it like a biological GPS. Its only job is to find the exact spot in our DNA that we want to edit. It brings along a partner, an enzyme called Cas9. Second, this Cas9 enzyme, those are our molecular scissors, makes a clean slice right through the DNA strand. And third, and this is the beautiful part, the cell's own natural repair kit kicks in to patch up that break. The system basically uses the body's own tools to make the final change.
But here's where the real power comes in. It's what happens during that repair step. This is where scientists can really work their magic. They can guide the outcome in one of 3 ways. They can just disrupt a faulty gene, kind of turning it off. They can completely delete a harmful sequence, or, and this is the absolute game changer, they can give the cell a new, correct piece of genetic code to use as a template, basically fixing that original typo we talked about.
Now, I want to be really clear. This is not just theory anymore. This isn't just something happening in a lab. The technology has officially made the leap from research to reality. Let's talk about the very first CRISPR cure that's been approved.
That's right. As of late 2023, there is officially one FDA-approved therapy that uses CRISPR. It's called Casgevy, and its approval is, without a doubt, a historic milestone for medicine. This is that moment where a revolutionary idea becomes a real-world treatment that can actually change patients' lives forever.
So what does it do? Well, Casgevy is designed to cure 2 really devastating inherited blood disorders, sickle cell disease and beta thalassemia. The way it works is just amazing. They take a patient's own stem cells out of their body, edit the faulty gene, and then put the corrected cells back in. For people who've spent their entire lives dealing with debilitating pain and constant blood transfusions, this is, for all intents and purposes, a functional cure, all thanks to one single precise genetic edit.
But you know how science works, right? It never just stands still. So even as this first CRISPR therapy is just now reaching patients, researchers are already hard at work on even more advanced, even more precise versions of the technology. And the analogy for this next step is just perfect. If the original CRISPR is like a pair of scissors that just snips the DNA, these next-generation tools like base and prime editing are much more like a word processor, like using a pencil and an eraser. Instead of making that big cut across both strands of DNA, they can go in and chemically rewrite single DNA letters directly. It's a much more subtle and precise process with fewer potential mistakes. And to take it to the next level, scientists are now pairing these tools with artificial intelligence. AI models can crunch through enormous amounts of genetic data to help design the absolute best, most effective, and safest guide RNAs. This helps minimize risks and really speeds up the whole process of developing new treatments.
Okay, but with this kind of incredible power comes immense responsibility. It is absolutely crucial that we also talk about the challenges and the really profound ethical questions this technology brings up.
So the primary concerns really fall into a few big categories. On the science side, there's the risk of off-target edits. That's when the molecular scissors accidentally snip the wrong part of the DNA. But then you have these huge societal questions. How do we make sure these life-saving and, let's be honest, very expensive treatments are available to everyone who needs them and not just the rich? How are we going to regulate this thing to ensure it's used safely and fairly all around the world?
And that brings us to a really, really important distinction. See, all the therapies we've talked about so far only affect the individual patient, but there is a very clear ethical red line that the entire global scientific community agrees on. We do not cross into germline editing. That means making changes to DNA that could be inherited by future generations. For now, that is strictly ethically off-limits everywhere.
So even with those big questions looming, the potential here is just breathtaking. Let's look ahead a little and see what might be just around the corner as this technology keeps getting better and better.
The future is just incredibly exciting. I mean, researchers are actively looking at CRISPR-based treatments for a massive range of conditions. Everything from different types of cancers and high cholesterol to HIV and even neurodegenerative diseases like Alzheimer's. And this is where you really see different technologies coming together. We've got AI guiding the edits, and we've got nanotechnology being developed to create tiny delivery systems to get these tools to the right cells more effectively. It's amazing.
And that really brings us to our final thought. For the first time in history, we have a tool that can fundamentally change the code of life itself, a tool that could cure diseases that have been with us for millennia. So the question for all of us isn't really, can we do it anymore? It's how should we do it? As we stand here at the dawn of this brand new era in medicine, the most important question is, what will we choose to write?